Now remember we talked about the aspect of magnification, and where we were talking on the aspect of using the scale bar. Now after you have measured the length of the chloroplast in millimeters, right, you can convert it to micrometers. Then you should find that it's around 80,000 micrometers long. Then you calculate the real length using the formula, actual size of the object, then which is equals to size of image divided by magnification. We found out that the magnification is 100,000, right, and then is around 10,000, and then the size of the image is 80,000. So we divide 80,000 by 10,000, which gives us 8 micrometers. Then you've now found the measurement of the chloroplast. So in the first example, you were looking at the actual size of what? Of the micrometer, of the mitochondria, where you've been given the magnification only and then the cell itself. So you are to measure the length of the cell, then you are converting it into micrometers, then you divide the actual size of the object. But there's a scenario where you are given the length of the scale bar, and you are going to first find the actual magnification of the cell, and then you convert that size into micrometers. Then you are able to then find the actual size of the object after you found magnification. So there's a step where you find first the magnification of the cell. After finding the magnification of the cell, then you can convert that and calculate using the formula for the actual size to find the real length of the cell. So this is where you are dealing with magnification. So you can also be exposed to measuring cells using a graticule. So an eyepiece graticule is a little scale bar that you can place in the eyepiece of a light microscope. When you look down the microscope, you can see the graticule as well as the specimen. So the graticule is marked off in graticule units. So you can use the graticule to measure the specimen you are viewing on these graticule units. Just turn the eyepiece so that the graticule scale lies over the object you want to measure. Now, the graticule units have to be converted to real units, such as millimeters or micrometers. So this is called calibration. To do this, you use a special slide called a stage micrometer that is marked off in tiny scale. There should be information on that slide that tells you the units in which it is being marked. The smallest markings are often 0.01 millimeters apart, and that is 10 micrometers apart. Take a specimen off the stage or microscope and replace it with the stage micrometer. Focus on it using the same objective lens as you used for viewing the specimen. Line up the micrometer scale and the eyepiece graticule scale. You can do this by turning the eyepiece and by moving the micrometer on the stage. Make sure that two large markings on each scale are exactly lined up with each other. You should be able to see the way the arrangement where you find eyepiece graticule and the stage micrometer scale. So you can see that the markings on the micrometer can be lined up with the markings on the eyepiece graticule. So work towards aligning these markings so that you see another line lining exactly with the other. So work along towards the right until you see another line, another two lines that are exactly lined up. There is a good alignment of where you find a good alignment on the stage micrometer and on the eyepiece graphical, so that you are able to find, let's say for example, if there is a good alignment of 68 on the stage micrometer and 9.0 on the eyepiece graphical, you can be able to say 8 small eyepiece graphical markings equals to 18 stage micrometer markings, which is equals to 18 times 0.01, right, millimeters, to convert it into 0.18 millimeters, and this gives us 180 micrometers. So one small eyepiece graphical markings, right, is equals to 180 divided by 80, which is the 80 small eyepiece graphical markings, which gives us 2.25 micrometers. Now you can be able to calculate the real width of the plant cell that you have measured. So if, let's say for example, it was 23 eyepiece graphical units long, so that then its real width will be 23 times the 2.25 micrometers, which is the small eyepiece graphical marking that we have found, which gives us 51.75 micrometers. So if you want to look at something using a different objective length, you have to do the calibration of the eyepiece graphical units all over again using this length. So once you have done it, you can then save your calibration for the next time you use the same microscope with the same eyepiece graphical and the same objective length. So this marks the end of the first topic, where we were dealing with the cell structure, and we are looking mainly on the microscope. And remember, in summary, we talked about the light microscope, how the light microscope uses a light rays that pass through the slide and then are focused on objective length, and how the electron microscope uses the electrons rather than light rays. And you should be able to define magnification, where we say that magnification is equal to size of the image and actual size of the object. Then you can rearrange this as actual size of an object, which is equals to size of image divided by magnification. And so you should be able to calculate magnification and also find out how to make use of the graticles. So in the next session, we are then moving now to the cell structure and function. So follow our Facebook page, and also follow us on YouTube to receive these online tutorials. Thank you.